first presentation is about the analytical method lifecycle management. And this is focused on a new proposed general chapter in USP, United States Pharmacopoeia, General Chapter 1220. But I will be providing you with compendial perspective. So, but before I make any of my presentation, I would like to make a disclaimer that the presentation that I'm about to give you are my views and should not be construed as views or policies of the United States Pharmacopoeia, US FDA, or International Conference on Harmonization. In terms of the outline of my presentation, I will first discuss some of the references because this new proposed general chapter in USP came in as a stimuli article. Also, afterwards, before I actually go into the details describing you this general chapter, I will provide you with some background in terms of the current analytical method compendial management. It's a long title, but these are the processes and challenges of implementing new method in a laboratory. I will then move on and describe to you the proposed USP General Chapter 1220 Analytical Procedure Lifecycle, and discuss with you the potential changes in terms of compendial standards. Finally, at the end, I will try to summarize and provide you with some of the advantages and possible changes. So as an introduction, let me just ground everyone in terms of USP, United States Pharmacopoeia. So only in United States is where the FDA is separate from the Pharmacopoeia, because the US Pharmacopoeia was established 200 years ago, way ahead of the US FDA. But the US FDA uses the standards that the USP creates to ensure the quality of drug products in United States. And I believe many other countries, including the Philippines, uses USP as part of their standard setting for drug products in the Philippines. So in the book of USP, there are three important things that you need to know. One would be the general notices. So the general notices in USP contains requirements that are applicable throughout the book of USP, but it is superseded, meaning a more detailed instruction is given through what is called the general chapter or monograph. So in the general chapter in USP, these are the requirements that are applicable to many of the monographs. When we say monographs, at the end, I'll describe to you what it is, but the general chapter supersedes the general notices. And general chapters in USP are referenced in monographs. If they are numbered below 1,000, they are termed enforceable chapter. If they are numbered above 1,000, those are general chapters that are informational chapters. General chapters can be procedural, which means they tell you a procedure, a test method, but they can be a guidance general chapter. And finally, we go down to the one where it supersedes both the general notices and the general chapter, which is the monograph. So the monograph contains the requirements that are specific to the article. When I say article, it's referring to the drug substance or active pharmaceutical ingredients, API, or the drug product, or what some term as finished dosage forms. So the monograph contains all the requirements and it supersedes the general notices and general chapter requirements. In terms of references, where before the new proposed general chapter 1220, it's called the Analytical Procedure Life Cycle, was published in PF, Pharmacopoeial Forum 43.1, there were publications that were published before it and stimuli articles that were published after it. So these publications that were published before are stimuli articles that have important terminologies. if you want to find out more about the new General Chapter 1220, you can read more some of the background information that were published before as listed in this slide. So it contains method development procedure, performance qualification, performance verification, and then you have the target measurement uncertainty, the analytical target profile, and the analytical control strategy. Afterwards, there were also stimuli articles that were published after the publication of the PF 43-1, which is proceedings of the workshop and also the measurement uncertainty for the pharmaceutical industry. So I apologize, some of the jargons may be not familiar to many of you because I have the audience have a varied experiences, but you can always send your questions at later time, okay, after my presentation, and we can answer some of those. The goals of the analytical lifecycle management is that the different general chapter guidances that USP provides in terms of the validation, verification, and transfer of analytical procedures were written at different times, and they were done separately from one another. So they are not very much well integrated, and I will show you why. So therefore, there's a new way of working that has been proposed, which is this General Chapter 1220, that is more effective and efficient through an interrelated and a continuous process. Also, the increasing the knowledge of the procedure that contributes to the different sources of variability for a method will result in fewer failures during qualification or verification. And then the design of more robust procedure or method will also result in fewer failures such as system suitability. And finally, if there is an increase in the facilitation of continuous improvement and change control, will provide a better way of working in terms of method development and validation in a laboratory. Utilization of new analytical method versus traditional HPLC. So I've been in the pharmaceutical industry or in as an analytical chemist in the United States for more than 30 years. And it's been HPLC, high performance liquid chromatography, that has been used really as a workhorse. Although there are other new sophisticated instrumentations available, but industry-wise, in a practical sense, it's always the traditional HPLC. So the production of pharmaceutical may be changing in the future, such as the continuous manufacturing. That's a terminology. So a new analytical method would be something that is going to be useful. Now, the analytical lifecycle management may result in the additional effort in terms of understanding the guidance and regulations. So the guidances are more on the regulatory and regulations, adapting new ways of working. So in a setting, in an industry lab, for example, or even in regulatory lab, always a method goes through a method development and validation. It may change based on this new proposed general chapter. And then finally, acquiring the knowledge and skills that is required to comply. With this new proposed general chapter, there may be requiring people to know more about statistics, design of experiments, quality by design, and risk assessment of different situations. So the approach for this new proposed general chapter 1220 is holistic, meaning it is covering all the different activities from method development to validation, routine use, change control, until the method is retired. And this enhanced approach improves the method understanding and performance that is leading to, first, fewer out of specification results. You guys in the industry knows what is this out of specification results. It facilitates method transfer. So you're moving one method, one procedure from one site to another. And also the potential to lessen the regulatory burden. So this is something that is very much of interest in the United States because the FDA really goes after the industry in terms of how they manage their product. So before I describe to you the General Chapter 1220, what I wanted to do is to provide you with some background in terms of current implementation of new analytical method in a laboratory. So if you're, for example, an industry, a pharmaceutical industry, you are using different methods to analyze your sample. Your methodologies can either be coming from the USP method, which is the book. It's termed Compendial. But at times, there are methods that are not in USP. So those are termed non-compendial or alternative methods. Those are the internal methods from your company. However, this will have to adapt into the new proposed General Chapter 1220, starting with the establishment of what is called the Analytical Target Profile, ATP, and Target Measurement Uncertainty, TMU. These two parameters is consistently the standard in the three stages of the procedure life cycle, starting with stage one, which is procedure design and development, having 5 different elements. Stage two, which is procedure performance qualification, having 3 elements. And then finally, stage three, which is termed as the continued procedure performance verification. Again, it has 3 elements. These 3 stages, stage one, two, and three, are all interconnected with each other so as to provide a continuous process of improvement. So it's not one way, but it could cycle back and forth. So we go to the current implementation of new analytical method in a laboratory, either by using a USP method or an alternative method. This involves actually examining or having an understanding, for most of you who are not familiar in the pharmaceutical industry, uh, goes from the drug development, starting from drug development, going into the preclinical testing, and then to what is called the investigational new drug, and then FDA reviews them. And it goes to the 3 different clinical phases, phase 1, 2, and 3, until it becomes a new drug application. It's continued on in terms of the review from the FDA until the analytical method is finalized before the company gets approval for marketing. But once the drug is in the market, there may be some changes in terms of the manufacturing after post-approval changes. Now, United States Pharmacopoeia is focused when the drug is in the market. That is, we set the standards for those pharmaceutical products that are in the market. So industry-wise, the pharmaceutical industry is focused in terms of regulatory requirements, starting from the drug development all the way until the drug is approved. So I wanted to make that clarification between the regulatory FDA and the compendial USP. So just as a summary, a method typically goes from the development stage to validation. So the validation guidelines based on USP is General Chapter 1225. So this procedure or methods are routinely used in a laboratory. Either there is a need to do a verification of the product being produced, or there is a need to transfer the analytical procedure, for example, from a research lab to a manufacturing site. The USP General Chapter Guidance created at different times to address the needs of the customers, but they are not well coherent, as I mentioned earlier. So those 3 general chapters, starting with the USP General Chapter 1225, describing the validation of compendial procedures, is heavily focused on chromatography, I mentioned earlier, and was created before the ICH-Q2, which is a description of how to do method development and validation. ICH, by the way, is International Conference on Harmonization, and it is more of the international standard for drug products and drug substances. So it covers the different countries. So that's more of I call United Nations, but that covers the major USP, British Pharmacopoeia, and also the Japanese Pharmacopoeia. Now, USP General Chapter 1226, which is the verification of compendial procedures, were created to support the needs of the stakeholders, and the General Chapter 1224, which is the transfer of analytical procedure, is the one that has been recently added as a general chapter, because for many years, organization just move one method or procedure from one site to another without much formal process. So what is the process of implementing new method in a laboratory, either compendial or alternate or harmonized? So this slide, this is next slide, will describe to you the process. So when you are implementing, for example, a procedure coming from the USP book, which is termed compendial, and you're analyzing your product, you must do verification. But this is only the first time, one time. When you're doing verification, you need to do documentation and determine if the method is suitable to analyze your sample. If it is suitable, then you can do implementation. But if it is not suitable, and because you are using, for example, an HPLC method, you can use what is called the USP General Chapter 621, or the System Suitability Adjustment. So you can make those adjustments and determine if the method is appropriate to analyze your sample. If it is suitable, then you can do the implementation. And if not, then you'll have to contact United States Pharmacopoeia for possible alternate method or procedure, or even the creation of what is called a flexible monograph. Now, if you are implementing an alternative method, which is termed internal to your company, it is important that these methods and procedure must be validated so as to be able to do any transfer to one side to another. If they are not validated, then you need to validate them based on the General Chapter 1225 method validation. I will discuss this at the last presentation. And in terms of transferring, there are 4 different ways of transferring an analytical procedure, but they are again based on whether the method is validated or not. If the method is validated, for example, and everything is the same, meaning the instrumentation, the staff, and the procedure doesn't have much issues, then there's what is called the transfer waiver. So you can just transfer it without much documentation or experimentation. If not, then you can analyze a common sample between a transferring lab and a receiving lab, and this is termed as the comparative testing. And if not, if you still have issues, you have to do what is called the revalidation, meaning the receiving lab has to do or some or most of the validation processes. If the method to be transferred is not validated and the transferring lab is available to do some of the validation, then there's the 4th option called the co-validation. So this is a collaboration between the transferring and the receiving lab. So these are the 4 options in terms of a formal movement of a method or procedure from one site to another. So what are the ways to deal when you come up with issues implementing a new method or procedure? So when I was working as a scientist at United States Pharmacopoeia, I do get some calls from the industry in the Philippines asking questions about some of the difficulties that they have. Just to clarify, if you are having difficulty and you're using HPLC and you're using a USP monograph, you can use what is called the USP General Chapter 621. But this, again, is only applicable to USP monograph. If not, if you're using an alternative or harmonized method, one thing you could do is to make the adjustments. But remember that these methods that are not reflected in USP monographs are subject to regulatory review. So FDA can ask you questions about the validity of your method. So these are not reflected in USP monograph. The third way would be If you want, you can contact USP so that your modified alternative procedure can be added to the USP monograph, simply because you have a little bit of a different product compared to the other products. But this would require a lot of collaboration with USP staff. So these are the ways to implement new methods if you're having issues. Now, this slide is a summary of what is called the System Suitability Adjustment Guidelines for HPLC. Now, the first column lists all the variables in HPLC. The second column are the allowable range of adjustments. So you can make adjustment changes. If you are using a USP monograph, you can apply these allowable adjustments. And the nice thing about it is you do not have to do any validation. You can apply, and then if it works, then you're fine. Keep in mind that these allowable adjustments are limited to whether your system is isocratic or gradient. Some of them allows either isocratic or gradient, but some of them are only limited to isocratic. So when we talk about isocratic, meaning it's one mobile phase in HPLC. If it's gradient, the mobile phase is changing. So anyway, this is just a summary of what is available for the USP General Chapter 621. Also, keep in mind that, hold on. I guess I cannot go back. In the previous slide, keep in mind that, thank you. Keep in mind, if you are using other pharmacopoeia, for example, like the Japanese pharmacopoeia or British pharmacopoeia, the adjustments for system suitability are different from USP. Japan has their own adjustment guidelines, and British pharmacopoeia have their own adjustment guideline. Thank you. So let me go to the next set of slides, which is discussing the analytical procedure life cycle. As I have described to you, the implementation of a compendial or non-compendial methods in the laboratory, I mentioned earlier that these have to be adapted into the new proposed life cycle, starting with the analytical target profile, ATP, and also target measurement uncertainty, going to the 3 different stages, stage 1, 2, and 3, which are all interconnected to each other for to provide a continuous process of improvement. Let's talk about the analytical target profile. Analytical target profile states the required quality of the results produced by the analytical procedure in terms of acceptable error in the measurement. Also, it states that the allowable target measurement uncertainty associated with the reportable value. And then because of the ATP describes the quality attributes of the reportable value, it is applied during the procedure life cycle and connects all the 3 stages. I mentioned that, stage 1, 2, and 3. And then the ATP criteria are derived from external requirements and not only from the performance of the analytical procedure. If you are familiar with method validation, you understand the use of what is called the analytical performance characteristics, the accuracy, linearity, limit of detection, so on. But those are what is termed as internal requirements. The ATP is derived also considering the external requirements. The external requirements, as when we discuss again verification, those are focused on the method and the sample. Those 2 things, method and the sample. The acceptable level of risk in making an incorrect decision is considered when establishing ATP, which means you need to have some level of understanding in terms of statistics. And then the reportable value may be the mean of multiple analytical results. So A replication strategy, number of trials have to be defined in the analytical target profile. So when you're establishing the ATP analytical target profile, it's important to consider sample to be tested, matrix in which the analyte will be present, the allowable error for the measurement as assessed through the accuracy and precision. These are the ones that make up the target measurement uncertainty. Also, the allowable risk criteria that are not being met. And then finally, assurance that the measurement uncertainty and risk criteria are met. So those are the things about the ATP. We go into the target measurement uncertainty terminology. TMU is the maximum uncertainty that can be associated with a reportable results while still remaining fit for purpose. So TMU is a comprehensive term versus the traditional term for just the precision, which is representing random errors, and the bias, which is more on the accuracy. So TMU can be considered to represent all the associated factors that affects the ATP. So the two main elements in target measurement uncertainty would be the bias or accuracy and precision. So in this next slide is a consolidation of attributes contributing to the TMU in terms of the accuracy and precision. So list of accuracy is listed here. You have the selectivity, linearity, extraction, efficiency, and everything else. So also in terms of precision, those that needs to be considered would be sample preparation, weighing, instrument, integration, and so on and so forth. So you notice that these elements or these listed factors for both accuracy and bias and precision are not limited to only the internal to the method, but it is also external to the method, which covers both, again, the methodology and also the sample. Let me discuss with you briefly the three different stages. So in the first stage of the proposed general chapter, which is the procedure, design, and development, 5 elements, where the first one would be knowledge gathering. This would be the stage where you're trying to determine what kind of methodology, what kind of procedure am I going to use? Do I use HPLC or near-IR or Raman? The next step would be to find out the risk assessment, because after knowing all the relevant information in the first element, the knowledge gathering, then you determine what are the risks in terms of the variables that will impact my reportable values. You may be using what is called an Ishikawa diagram or the design of experiment tools. If you're not familiar, you can learn those things. The analytical control strategy would be the third thing in this stage 1, which is you are going to be establishing a planned set of controls derived from the requirements for fitness for purpose, meaning what are your control strategy so that you can maintain the ATP, analytical target profile, and the target measurement uncertainty. Fourth one would be your replication strategy. Again, this is trying to cover the statistical analysis of the samples. The last element in the stage 1 would be the knowledge management. This would include acquiring, analyzing, storing, and disseminating information related to the activities involving the development, technology, transfer, qualification, and change management. So this is something important also. After you complete the stage 1, it is good to evaluate the data that you obtain in terms of the absence of significant bias so that your precision is at the appropriate level. And then before starting the qualification, which is stage 2, you can perform a compendial procedure from other pharmacopoeias such as Japanese pharmacopoeia, EP, BP, other than USP. Also, you can perform what is called a quality risk management. And then the third one would be to verify control strategy of the procedure so as to ensure that the analytical target profile are met. So when you start stage 2, which is the procedure performance qualification, it has 3 elements. The first one is to establish a protocol. What are those protocols? Protocols are those that contain the ATP, the method-specific performance, the description of the procedure, description of the experiment, test samples, all the statistical approach to be used for data analysis. This is followed by the qualification, study design, and execution. This step actually confirms that the procedure generates the reportable value that meets the ATP criteria and that it remains appropriate for the testing of the product in the environment that it will be used. So remember, the environment is actually the method and the sample. And then finally, the third element in stage 2 is to write a final report. So after completing the stage 2, one would need to refine and update the analytical control strategy. Because, for example, this would include, let's say, you wanted to have additional control so as to reduce the sources of your variability in a routine operation, and you can modify the replication strategy or replication level. Say, you can increase the number of injections, you can increase the number of sample preparations, and these are all based on the uncertainty of the reportable values. When you start the stage 3, it has its term as the continued procedure performance verification. It has 3 elements. The first one would be the routine monitoring and trend analysis. So during this time, it's important to determine I'm sorry. Was I muted, Chris? Chris,
Okay, can go on po.
can I continue?
Yes po, yes po.
Okay. Okay. So continuous improvement would be the second element. So as after determining the different out of specification or out of trend investigation, the continuous improvement can ensure that the analytical procedure remains in control, and then a change control is needed at the end so as to ensure that the level of activities confirm the level of change in producing fit for purpose in terms of risk associated with change and knowledge available about the procedure and the effectiveness of the control system. In terms of the potential changes in compendial standards, I'll provide you with an example of the current assay procedure. I'll also provide you with how additional control ranges can be included in the monograph, and finally, the addition of analytical target profile. So this is an example of a current assay procedure from USP, where I put together the sample and standard solution and the chromatographic system, the mobile phase, system suitability, analysis, and acceptance criteria. So addition of control range would be good to add in monograph. For example, in this example that I gave you, the sample and standard solution can have a table that contains parameter. For example, it can have the solvent composition, mixing time, and heat on steam bath, and it would have a target value with a low range and upper range. Similarly, in the chromatographic system, it can have several parameters that would have a target value with a low range and an upper range. Addition of analytical target profile is described in this next slide, where the results are shown, the 4 possible outcomes of reportable results and the expanded uncertainty as described by the Gaussian curve are compared to the specification. So if your specification is the upper limit, there are 4 possible results, where the first one would be a result that is above the limit. The second is the results is above the limit, but the limit is within the expanded uncertainty. So you can see with this intersection between the Gaussian curve and the upper limit. A third result could be the results is below, but there is also possibility of the limit is within the expanded uncertainty. And the fourth one is the result is below the limit. So the question is really how much overlap is acceptable between these 2 situations. So an example of assay procedure with analytical target profile is shown here. The analytical method is capable of quantifying metoprolol target in metoprolol tartrate tablets from 90% to 110% of the true value with an accuracy and precision such that the results reside within not more than 3% with 95% probability. Also, to include in the analytical target profile, this ATP for the content of metoprolol tartrate in metoprolol tartrate tablet is included in this monograph for the convenience of users for which the following procedure conforms. And finally, if you have an alternate procedure, it would say that the alternative procedure should be demonstrated to conform with these requirements. So in summary, this new proposed general chapter is focused on the future trend of the analytical procedure development and quantitation and continued monitoring. So advantages of this approach, again, is to have an improved understanding of the procedure and control the sources of variability. It will produce a more robust procedure. There will be a reduction of overall resources that is required to revise the procedure, and also identification of any adverse trend and continued improvements. Possible changes in terms of USP or compendial requirements would be the information regarding the method development may be required when submitting your dossier to USP. There may be an increase in the use of non-conventional procedures. These are what is termed as the multivariate near-IR or Raman spectroscopy. There may be a reduction in terms of the frequency of monograph revision. Enhance the use of quality by design, QBD approach, and finally, minimize the challenges in the compendial procedures for especially the OTC products in the United States. So these are some of the difficulties, and I can discuss this at a later time if you are interested in the challenges for the OTC drug products in the United States. But at this point, I thank you for your attention.